So just north of us in Austin is our wholly owned subsidiary, Signature Science LLC, which, much like SwRI, focuses on research and development that solves scientific and technological challenges. For our next presentation, Dr. Anthony Capel describes a project for the Centers for Disease Control and Prevention to develop a new workflow for identifying RNA viral threats, enabling researchers to quickly identify and address new and emerging hazards. Anthony.
Thank you for the introduction. I'd also like to thank the organizers for having me speak today about some exciting things that are happening at Signature Science. I'm a molecular biologist. Here's a simple and abbreviated view of molecular biology. Genomes are the blueprints for organisms. You, me, plants, animals, bacteria, and viruses. The majority of these genomes are made up of deoxyribonucleic acid, or DNA, and that's pictured here on the left. It has a classical helical structure and is called double-stranded, meaning that it's kind of like a photo and it's negative right next to each other, so it has the same information locked in different forms. Luckily for us, scientists and engineers have developed ways of reading this DNA, or sequenced DNA. We simplify this into its four basic letters of the unique chemicals that make up DNA as G, C, A, or T, and you'll probably see that during my talk. Now, another important step that happens in our cells is transcription. It's the process in which portions of this DNA of our genome get copied into RNA form, and that's how you get different tissues such as blood, skin, muscle. And RNA is ribonucleic acid, and that can make up the genomes of viruses as well. So they don't have DNA, but RNA viruses have RNA genomes. RNA is actually very hard to read, and so we need an additional step to be able to read RNA, and that's known as reverse transcription. And this is harnessed from RNA viruses as well, where RNA is converted into DNA. I should also mention that RNA is a single-stranded structure, so there's no copy of that information, and so you can get errors at a much higher rate than you do with DNA genomes. I should also mention that just despite being able to read RNA and DNA, we don't necessarily know, based on the genome sequence, if it's harmful in any way. So like questions such as, can it kill us, can it infect us, are not readily answerable by sequence alone, and we're kind of interested in trying to approach that as well. RNA viruses are a global threat. You're probably very well SARS-CoV-2, the severe acute respiratory syndrome virus that has been causing this COVID-19 pandemic for a while, and it's very multiple variants of its RNA form. You also probably are aware of Marburg and Ebola viruses, and where the infected die at about a 90% rate. We also have a family of viruses that you probably heard in the news: Zika, dengue, West Nile, all carried on insects such as mosquitoes and ticks. And along with some other very well-studied viruses, these seem to be emerging and re-emerging every year with slightly different RNA genomes. There's also those unknowns, the ones that we don't know are out there that can escape the current state-of-the-art detection methods. And that state-of-the-art current detection method is known as polymerase chain reaction, or PCR. You may be familiar with this as well because you probably had a COVID PCR test to get on a plane during this pandemic. PCR takes a small part of the genome as a target and amplifies it hundreds and thousands of times. And so what you get is something that's easy to detect and easier to study. But PCR only works with DNA, so you still have to do that reverse transcription step, converting RNA, such as from the SARS-CoV-2 virus, into DNA, even before you can do a PCR. And that can be time-consuming, and it can fail. There are three current challenges to RNA viral detections in a biothreat event or in emerging infectious disease, and they're widely different. They have different sequences. Their genomes are just so variable. And plus, they change rapidly because they are made of RNA. They're more likely to get errors. PCR relies on them being stable. So it's possible that state-of-the-art detection through PCR can fail through small changes within the sequence of RNA viruses. Even to develop a PCR, you have to know the sequences of these viruses to actually identify what target can be unique. Viruses also need other cell machinery to grow and to replicate. So they need to infect something to replicate. So to study viruses such as this, you need to identify a cell culture in which it'll grow. This can be very difficult because they're very picky on what cells they infect. And of course, you need reverse transcription, which makes DNA from RNA for any analysis to work. And because we're really good at reading DNA, we kind of need to do this. So what we've developed is a workflow using an untargeted sequencing method with a third-generation sequencing platform known as the Oxford Nanopore MinION pictured here. It really is handheld and attached to a laptop. So you don't need to know the sequence of what you're detecting because it's untargeted. It's very robust against mutation because you're just sequencing anything and everything that's there. You don't need to grow it, the virus, to detect it, so you don't need to grow it. But we still rely on this reverse transcription to convert RNA to DNA. And so that really kicks off our workflow. We've been focusing on respiratory viruses as well as blood-borne pathogens. So so it's the COVID test where it shoves a swab up your nose and puts it in VTM, and then we extract it. And then we have to do this reverse RNA step to get it. These samples that we initially tested didn't really have much material to start with, so you don't get much DNA material back. So what we needed is kind of like an untargeted PCR to amplify this. So we looked at whole genome amplification, and this process is very interesting. What it does is kind of just makes it's a huge copier, which copies the entire genome. While PCR, you can kind of think of as a targeted copier that only makes a 8x11 letter size copy of the genome. This is a banner size copy that just copies the entire genome. And not only does it make the copies, but the copies also lead to other copies. Another way of looking at this is if you're making spaghetti and you put one strand of spaghetti into the pot, and by the end of its cooking, you got a whole pot of spaghetti. And that's kind of what this is. You just have lots of material at the end. And this makes sequencing it a lot easier. And this also helped really increase our detection. You clean it up, you put some chemicals in that makes it able to go onto the Oxford Nanopore pictured here. This is known as a flow cell, and this is a drop of the library, which you put into it. It's known as the library because it's about to be read. Essentially, it's just a book of GCs, A's, and T's that you're about to detect. Now, the important part of the Oxford Nanopore technology is the nanopore part. This is pictured here. The DNA is here and attached to a motor. The size of this pore is so small that only half of the helix gets through. So it actually breaks it apart and then pulls it through. There's a voltage across this, and what happens is the DNA rapidly gets through. This actually tries to slow it down a little bit because you need to be able to detect what you're looking for. And it kind of creates a funnel. And when it gets to this point at the narrowest part of the funnel, only single parts of the DNA can get through. And this causes changes in the current. ONT, or Oxford Nanopore Technologies, calls these squigglies. It's their technical term. And that gets translated into the DNA sequence, G, C, A, or T, based on the squigglies. So you have the DNA sequence now. We use something that's been developed at Signature Science called S2Fast, which is sequences of functional analysis of threads. And that was part of an IARPA program known as FungiCat, IARPA being the Intelligence Advanced Research Project activity called FungiCat. And FungiCat stands for Functional Genomic Computational and Assessment of Threat. And I love how they incorporated GCAT in there, same as DNA. What this does is kind of tries to address those questions that I talked about at the beginning. Can you have a sequence and learn from it? Does it infect us? Does it kill us? And that's kind of what S2Fast is trying to answer. Now, it generally looks at everything as individual sequences and tries to rate it based on the potential of causing harm, such as, is it a toxin? isn't known to be in a pathogen, can it kill you? Part of this, so it's proprietary, and generally it's very intensive, but a part of this program is SeqScreen. And this is the kind of freely available, you can download it yourself if you wanted to look at sequences. And this is from our RICE collaborators who were part of the IARPA program. What SeqScreen does is it takes all the sequences off of the sequencer, kind of looks and see what's there, and determines what kind of database to make. So if it's something similar, it starts pulling reference sequences and makes this whole genomic reference sequences. It then pulls these and starts saying, hey, how similar are you to this reference? Now, I'm going to be very brief, but it does a lot of workflow with statistics and bioinformatics, so I'm going to try to simplify this down to, this is just a statistic that's generated by the software, and this is kind of our cutoff that we decided. That usually gives great results. So if it meets this criteria, that particular organism happens to be present. If it doesn't meet that criteria, it checks another statistic to see if it meets that criteria, and then it looks back at that old one and say, oh, well, yeah, that was probably there. And if it doesn't meet that, then actually it's probably something really, really close to that organism, but not necessarily that organism. And this is kind of where you start getting into those novel viruses that may be present. And of course, if it decides that it wasn't there in the first place. This is a real-world sample. This is from a remnant sample from an actual patient that had a respiratory disease that has been diagnosed. When we took it through our workflow and we used the normal bioinformatics workflows that we use today and the cutoffs that we use today, we got something below a threshold. It was definitely there. We could see it within the sequences. It's just it was not meeting the criteria of a detection. But SeqScreen actually detected 2 viruses. The first one, with the highest score here, matches the diagnosis based on PCR testing. So it detected it. We also have this other virus that wasn't even on the same PCR panel. So it's possible that they just detected the first one and didn't move on. That's generally what you do in clinical samples. But based on the patient information, it's possible that both were present, co-occurring in the same patient. So what this workflow has the ability to do is detect any RNA virus, known or unknown, in any sample, environmental, clinical. And while we focused on respiratory viruses as well as bloodborne pathogens, this can be expanded to many different matrices and samples. All the clinical samples and even environmental, such as mosquitoes, bats, pools of mosquitoes, such as they usually use for dengue and West Nile. But for the immediate steps, we have funding to transition this workflow to a new lab. So currently we're developing materials, training materials, and testing materials to transition this to a new lab. We're going to test it internally first, and then we have a partner in the LRN, the Laboratory Response Network of the CDC, in which they're going to take with it and try to run with it and see if they can do as well as we're doing inside of our lab. Finally, I'd like to thank everyone at Signature Science who's been working on this project hundreds of hours. This is funded through the Center of Disease Control. I should mention that any opinions, findings, and recommendations expressed in this presentation are those of me. I also like to thank IARPA, the Intelligence Advanced Research Projects activity that funded FungiCat, and our RICE collaborators that were on that project as well. Thank you.
Thank you, Anthony. Anybody have a question? Yes.
Yeah, I was wondering about the swiggle detection. Is that a voltage or absorbance?
So there's a voltage across the membrane between the pores, so it's a voltage change. And as it's going through, it causes current changes. And those squigglies are the current changes.
It's a very novel technology that Signature Science has been following for probably 10 or 12 years now since they first started. And it's a really amazing small device. One more question? Come on, no geneticists out there? All right, thank you, Anthony. Anthony will also be at lunch, so you can catch up with him. Thanks, Dr.